Hello, friends. This is Dr. Mack, Crayton III, Ph.D. Thanking you for joining me for another episode of the interface between science and scripture. Before we get started in this episode, be sure to hit the subscribe button that's below and also hit the alert bell so you'll be notified when new content is posted each week. You ready? Let's get to work. For our topic today, I've taken to the classroom to discuss evolution versus creationism. When it comes to arguments between these two groups, it often boils down to the molecular level, where evolutionists believe in a spontaneous random assembly of molecules that led to biomolecules and eventually living cells. Whereas creationists believe that all things were made by God and created with intelligent design. And this design has irreducible complexity. So let's get into it today. To bring in our science and spirituality, we go to the Holy Bible, the book of Colossians 2 and 16. And it reads, "For by him were all things created, that are in heaven and that are in earth, visible and invisible, whether they be thrones or dominions or principalities or powers. All things were created by him and for him." So when we begin to look at this verse, we see that God created some things that are visible, and we can see the birds, the animals, the trees, the earth itself, all visible. And then it talks about things that were invisible, and it begins to name spiritual things and things that may be mental and emotional. When we bring in science into the invisible with this particular verse, it may talk about cells or other things that are microscopic, meaning they can't be seen with the naked eye and need the aid of a microscope. So be it a cell, be it nucleic acids such as DNA, RNA, be it proteins, prions, viruses, all of these things are invisible to our eyes, but God created them all. And so we begin to look at things seen and unseen. So the question becomes, and has been posed for many decades and even centuries, as we see here on the screen, how did life begin on Earth, and how did the first cell arise on Earth? Well, of course, we're exploring the two different camps, creationism versus evolution, to see at the molecular level what's happening. So when we say the molecular level, we're down to the molecule. So we have the basic unit of matter, which are atoms, and atoms contain protons, neutrons, and electrons. Atoms will come together and join together using chemical bonds to form molecules. And these molecules, particularly those that contain carbon, are considered to be biomolecules or organic molecules. And these are the molecules that make up living things. So we want to begin to look at what are these molecules and what does science have to say and what does the Bible have to say. When we turn to science for the origin of living things, we have the RNA world hypothesis or theory that's been proposed and accepted in science. And according to science, the RNA world theory, the first RNAs were made using free-floating nucleotides that emerged in a primordial soup of molecules. They bonded together to make strands of RNA that were very unstable and degraded quickly. So for many years, as a student of biology, as a professor of biology, as a scientist, I've heard about this primordial soup. But I'm here to pose the question, exactly what is the soup? So was it some underground water source? Was the soup in glaciers or hot springs? Was the primordial soup the ocean itself? Where were these molecules floating around that came together? So that's something we often don't ask. What is the primordial soup? Next, to justify the simplicity and the primitive nature of these cells, scientists have said that the RNA that could have composed them, because RNA is a nucleic acid, as I mentioned earlier, the other nucleic acid is DNA. Well, RNA is a simpler molecule because it consists of a single string of a strand of nucleotides. So scientists propose that our genetic material might have been in those first cells RNA rather than DNA. But they state that this is a molecule that was very unstable and it could degrade quickly. Well, this is no different from what we know today. For those who are in biomedical research or scientific research using RNA molecules, they describe them as being transient molecules because of their instability and they degrade quickly. It's this characteristic from its beginning till present day that does not make RNA a viable molecule as the molecule of heredity. DNA, being a much stable molecule, can withstand the environment of the cell and conditions of the cell, as well as its transmission from generation to generation. So it's not a transient molecule, whereas RNA is. The other issue with the theory of the RNA world is that there has not been an organism, neither extant, meaning living today, or gone extinct, a prehistoric organism that is now extinct, that ever used RNA as its hereditary material. So to believe that these early cells contained RNA as a hereditary material, where there's no scientific record of any living organism ever containing RNA as its hereditary material, seems a bit implausible, right? The other piece in that is viruses, and this RNA world theory was likely based upon viruses. A virus can have RNA as its hereditary material or DNA, and we saw RNA viruses with coronavirus, right? And there are many other viruses that use RNA as its hereditary material. However, when it gets to biological sciences, viruses are not considered living organisms because they do not meet the 6 characteristics of living things. Now, that would be a different discussion to go through those 6 characteristics of living things. So you'll have to rely and hopefully trust me that viruses are not living when it comes down to that. As an argument against evolution and its spontaneous assembly of molecules. So let's dive into what is irreducible complexity. We look to these 2 different paragraphs. In the book, The Origin of Species by Charles Darwin, which was published in 1859, Darwin wrote, "It could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications. My theory would absolutely break down, but I can find out no such case." So what Darwin is saying here is, if you could find any organ, and at that time, we didn't know much about microscopic. We didn't have the field of, or the discipline of study called cell biology, originally cytology. These disciplines in biology, or subdisciplines of biology, came along after 1859, after Darwin had written this book. And so much of his theory of evolution was made without, or all of his theory of evolution was made without knowledge of cellular biology, which I am. As a geneticist, I am a cell biologist. So we then look to another professor, Michael Behe, a professor at Lee University. And so, we wanted to say, what is this concept of an organ that occurred? So what Darwin believed in natural selection, so that molecules would come together, and those that were better suited for the environment would survive, and that's where we get the survival of the fittest, and be able to move on or pass on their genes to the next generation. In this case, we're talking about random molecules coming together, and those that are being more successful, maintaining and formulating the first cell. So it's a little different from natural selection, where you have animals who are breeding and who are adapting to environmental conditions that allow them to breed with one another, and their offspring become successful for the next generation. So natural selection is a bit ahead of its time, or not necessarily appropriate to use when we're talking about the formation of the very first cell. Natural selection would apply when you already have living organisms. So Michael Bay believes he has found a system that was overlooked by Darwin, an irreducible complex biological system. If there was such a thing, would be a powerful challenge to Darwin's evolution. So exactly what is irreducible complexity according to Dr. Behe? Irreducible complexity is defined as the argument for irreducible complexity, IC, claims that some biological structures are too complex to be explained by known evolutionary mechanisms and therefore are probably the result of intelligent design, i.e., a creator or creation. So, what we're looking at is the cell itself and biomolecules. Biomolecules would be things like our nucleic acids, DNA, RNA. It would be proteins that we have. Other biomolecules that we have within the cell would be enzymes, hormones. We're going to look at phospholipids and so forth. We have four categories. There's carbohydrates, the sugars, right? Lipids, our fats, our proteins, and then we have our nucleic acids, are the four classes of the biomolecules. So we'll begin looking at, well, what are these biomolecules, and are they truly too complex to have spontaneously and randomly assembled themselves and then gathered together to form the first living cell? So let's take a look at that first living cell as proposed by science. Evolution believes there was a primordial cell, a primitive cell that existed or that came into existence from these molecules in the primordial soup. And so these molecules assembled themselves and we got the first cell. This is a diagram of that particular cell. Here we see phospholipids forming the cell membrane, and inside of the cell membrane we have RNA as its hereditary material rather than DNA. Now, the importance of a cell membrane is that it serves as the barrier. It separates the interior of a cell from the exterior of the cell. So a cell membrane is very important. It also serves as a protection or a gate, so it allows some molecules to come in, those that are needed by the cell, and then it allows excretion of waste, right? And other harmful molecules that may be too large to come across the cell membrane, it keeps those things out. So it's definitely a barrier to protect the inside yet from the outside environment. Now, what's not accounted for in that early primitive cell, or talking about that early primitive cell, is within the interior of the cell is the structure called the cytoplasm. And the cytoplasm is a complex network of microtubules and microfilaments. In this diagram, if you can see here, it's shown in blue. And so we're just going to omit that for simplicity's sake, since it's not addressed also by the evolutionists. What about the cytoplasm? And here diagram, they also have these little yellow spherical balls here. Typically that represents cellular organelles, but again, they're saying this cell was so primitive that it would not have cellular organelles. Cellular organelles would be little organs, and they serve the function as our organ system, a similar function as our organ system within our bodies. So it is a primitive cell. I will give them that. In comparison, I put in a picture of a diagram of our present-day cells. Prokaryotic cells and eukaryotic cells are the two classes of cells that we have. Prokaryotic cells, typically known as our bacteria, are more simplistic here, and they would be similar to the primitive cell that we saw over here. They have fewer cellular organelles. However, they do have a cell wall. They have DNA as their hereditary material, and there are other organelles in there. And here the name of some of the organelles that it has in common with eukaryotic cells. Eukaryotic cells are considered more advanced, and those are the cells that compose our bodies as animals, as well as plant cells are eukaryotic as well. And the difference is there is a cellular organelle called the nucleus, and the nucleus houses the DNA within the cell. So it's a membrane-bound organelle that houses our DNA. And so that would be characteristic of eukaryotic cell and true for both plant and animal cells. So we'll return back to our primordial cell and the two biomolecules that are named or prominent in that particular diagram, the phospholipids and the RNA. So here comes the reducible complexity. So we'll begin to look at the phospholipid. The phospholipid is under the class of lipids or fats. And in cells today, both prokaryotic and eukaryotic, our cell membrane is composed of phospholipids, and they are arranged into two layers, and therefore we call them a bilayer. We see that was the case in the primordial cell as well. So what is a phospholipid and its composition? If we begin looking at it, there's a head and a tail. The head of the molecule is described as a glycerol, the molecule of glycerol, and then there are two tails to the phospholipid. So here's our head and the two tails here. The two tails is described as a hydrocarbon chain. Hydro for hydrogen and carbon, the other molecule. So it's a string of hydrogen carbons joined together in this tail, and we have two of those. They are precisely made and always connected to the glycerol head to form a phospholipid. When we look at the chemical characteristics, the glycerol head is described as hydrophilic, meaning it has an affinity for water, hydrophilic, water-loving, and then the tail would be hydrophobic. It does not, it has an aversion to water. So when the bilipid arranges, the heads that may come into contact with the water is exposed to the exterior of the cell or the water that's on the interior of the cell within the cytoplasm. So again, we decided not to talk about the cytoplasm. And so the tails are arranged pointed towards one another, and they form the barrier that keeps the water that's on the outside, and the water and material that's on the inside of the cell on the inside of the cell. So that's the membrane barrier that it serves. So we can see the complexity of a phospholipid in itself. Remember, when we talk about the random assembly and spontaneous assembly, this molecule will have had to assemble itself in all of these different atoms that are coming together to form this specific molecule, would have had to come together and be able to do that repeatedly to form multiple phospholipids enough to form the entire membrane. Remember, for the cell, our cells are spherical in shape. For animal cells, counter-rectangular. Bacterial cells can have a variety of shapes. However, it would be a three-dimensional structure rather than just a little flat piece that we see here. Right? So that's a lot of phospholipids that would have had to assemble themselves spontaneously and repetitively to be able to form the cell membrane in just one cell. Right? When we look over here also, what do we know today from cell biology is that there's a cellular organelle within cells that compose or synthesize the phospholipids. And that cellular organelle is called the smooth endoplasmic reticulum. And so if that primordial cell did not, as the scientists would say, did not have cellular organelles, then the smooth, we call it SER, or smooth ER, the smooth ER would have been absent, and therefore we ask the question, how is this complex, or how was that complex molecule assembled? And so that's the irreducible complexity. Without the smooth ER, we don't have a mechanism to assemble the phospholipid. The phospholipid itself is complex in nature that you have to have the hydrocarbon chains and you have to have the glycerol head in order to have a phospholipid. Next, after generating a phospholipid, you have to have enough of those made to actually form the bilayer, and they have to be precisely arranged into a bilayer where the hydrophobic tails are contacting with one another, whereas the hydrophilic heads are orientated towards the outsides. So now we go to the next biomolecule that's named in that primordial cell, which was RNA. RNA, as I said, is a biomolecule that falls under the category of nucleic acids. So there's DNA and RNA, with RNA being the simplest of the two. However, it's complex in itself. It's just considered simple in comparison to DNA, which has two strands rather than just the one. So we began looking at our RNA, and the letters R and A stand for ribonucleic acid, and it's composed of its monomer units called nucleotides. According to the RNA world theory, these nucleotides, which have complex structures, it's made up of a base, a nitrogenous base, a pentose sugar, and one or more phosphate groups. So this is a complex structure, and there are four different types of nucleotides. There's adenine, guanine, cytosine, and uracil in RNA. So ACG and U in DNA is ACG and T. The thymine replaces the uracil. So we have four very complex molecules that would have randomly and spontaneously assembled into a nucleotide. And then from the nucleotide, each one of these would have bonded together to form a strand of RNA. That's a lot of randomness and a lot of spontaneity, right? For those molecules to have come together that way. What do we know about RNA today? We know that RNA is made actually from DNA. So DNA serves as a template for making RNA in the process of protein synthesis, right? Or transcription that leads to protein synthesis. So here's our DNA strand here. Here's an enzyme called RNA polymerase. So in order to make a strand of RNA, we need an enzyme called the polymerase, and there are other enzymes as well as like helicase. There are other types of polymerases. There's different polymerases that are in there. And you also have your proteins in there, DNA binding proteins and others to open the DNA and to allow these molecules to be added together. So there are enzymes and proteins that are necessary in order for the nucleotides to be assembled. Then it's a precise order. So the nucleotides are not just randomly put into an RNA molecule. They are based upon the order of the nucleotides that appear in DNA. And the nucleotides, which are in RNA, provides a specific message for making the protein or the amino acid sequence for our proteins. So it can't be haphazard because these are a set of instructions. It's the blueprint for making another molecule. So to say that these just randomly came together and that they could be in any order at any time is not feasible because you wouldn't have the correct code for making the necessary proteins. So we begin to look at this process of how these molecules came together. Even if the very first RNA molecule came together spontaneously, how could it reproduce itself? Otherwise, you would have a one-hit wonder, right? So we would need to be able to reproduce itself so that we could get progeny. There has to be other cells and other cells that came from the original cell, and there had to be a way for the RNA to be copied, to make copies of itself, right? And to do that. So today we would ask the question, how can we make an RNA molecule without its template, DNA? So DNA is our hereditary material and the blueprint for our bodies, right? And from the DNA, the genes in DNA, we make RNA, and from the RNA, we make proteins, and we describe proteins as the workhorse of the cell. So it gets things done, and so proteins can serve as enzymes as well. So we see this complexity here, and we talk about irreducible complexity. Without any one of these factors, the whole system comes to a halt. We often use the analogy, which came first, the chicken or the egg? How can you have an egg without a chicken laying it? And then how can you have a chicken if it didn't hatch from an egg, right? So it becomes a similar situation within the cell. How can you have these molecules if you didn't have the blueprints for those? Irreducible complexity. Last is the mention of reproduction. So as we talked about, whether it's an RNA molecule or a DNA molecule, These original cells would have had to reproduce in some way so that the waters would be filled with microorganisms that would give rise to larger organisms. So how would that division process occur? So here we have diagrammed some of your simple forms of what we call asexual reproduction. And this would be binary fission, which is typical in our prokaryotic cells today, such as bacteria, where the organism is just simply splitting in half. Before it does that, it copies its DNA so that the offspring all have a complete a complete set or complement of the DNA to start its new cell, the daughter cells. Another form is budding, which is common in yeast, and we know we use yeast for making beer or baking bread. Right? Their form of reproduction is budding, where a portion of that organism, this is the hydra, and it also does budding, and so it will have a portion that will stem off or bud off and then break off and grow into full size. Again, the full complement of DNA is contained within the bud so that it has a self-sustaining organism as its offspring. And the last fragmentation, such as with a starfish, where one of the arms or tentacles of the starfish would break off, and that small piece will develop into a full organism. Again, having the full complement. When we say having the full complement, again, we get back to the process of how do we make a copy of the nucleic acid so that the next organism can have the genetic material to sustain life. So it brings us back to the argument, irreducible complexity and evolution. With irreducible complexity falls under creationism and intelligent design. So based upon the complexity of biomolecules and the synthesis of a biomolecule, how could they have just randomly assembled into a cell and into themselves and become a living cell? It appears that there had to be some type of intelligent design, and that the intelligent design was from God, our Creator. Next, we have evolution and the primordial soup, where we go back to the notion of what was the primordial soup, and what's the probability, if we could calculate the probability of these molecules randomly and spontaneously coming together. What is the probability that that phospholipid membrane would have formed around a molecule of RNA that was just floating in the water to form the cell? Because from the indications and the structure of the primordial cell, there was no gated potential. We know today that the cell membrane has protein gates to allow a larger molecule to be transported across it. Right? So there were no gates and there were no mechanisms present to get that RNA molecule inside of that very first ball of phospholipids. And so we have to look at how did that randomly assemble together, and did all of these random assemblies happen simultaneously to come about with a cell? That doesn't necessarily follow the laws of science, right? The natural law. And then natural selection. Natural selection is applied to living things, how we adapt to our environment, and the strongest within that environment get to mate and pass on their genes into the next generation. And those genes become fixed into the population and become predominant. So natural selection is real. It's a way for diversification of organisms after we've gotten here on the earth. Why do we have different big cats? Why do we have different types of apes or chimps or the like, right? And so we have the diversification of animals based upon natural selection, adapting to a particular environment in which they live in. But it doesn't explain how the original cell came to be on the earth. So those are a discussion of evolution versus creation, irreducible complexity versus random association, random and a spontaneous association, or random and spontaneous assembly of molecules. So I thank you for the privilege of your time today. And I hope that you were enlightened by the discussion on today. If so, be sure to hit the like button for me and be sure to share it with others who might be interested in this same topic. If you have not done so, be sure to subscribe to the YouTube channel. Hit the alert bell so that you'll be notified when new content is posted. Remember, I love you, but God loves you more. Have a blessed day.